Hello friends. Today I'll be talking on very interesting topic, specification. And this is a demo session. So I have to give you very important points regarding specification. Okay. So let us understand the definition. Specification contains all the test parameters, then which procedure you have used for analysis. So you are giving the reference. Is it Pharmacopeial, in-house, which Pharmacopeia? And what is the method number? That is called as a reference. And the limits or standards which you have applied for that particular test parameter. So this as a whole is called as specification. Now, just look at the table. As an example, I have listed only one parameter, that is impurity E. So how it is presented? Under test parameter, impurity E by HPLC, limit or acceptance criteria not more than 15 ppm parts per million. This is a limit. And the reference of procedure is EP edition 10 European Pharmacopeia, and test method number is 010. So this way all other tests would be listed under specification, and this table would be completed, and that would be called as specification. Okay. Now, you people are familiar with this slide. You know all the pharmacopeias. So when you are preparing specification, the basic reference you will be using, that is called as primary reference, and these pharmacopoeia, they are the primary references to prepare specification. Yes. Now, if you want to sell your product or your raw material or an API in US market, then US FDA will expect you to comply with USP. If it is for European market, then compliance with European Pharmacopoeia, Farm Europa, and for Japanese Pharmacopoeia would be essential. So country-wise compliance is the first reference. Okay, so let us understand one monograph, isoniazid, which you people are familiar with, anti-TB drug, and I have taken it from EP 10 edition. Only tests are listed here. Okay, so here 11 tests are listed and they are extracted from EP 10. Now the question is whether only monograph compliance is enough from regulatory perspective. Very important question. So if I am listing these 11 tests and their limits and reference to analytical procedure, whether my specification is adequate from regulatory perspective, or agency is looking for something else. So the answer to this question is no, which is already given here. Now the reason is specifications are critical quality standards. So every company will design their own specification based on the criticality of that particular substance or product. Now, what are the references used to prepare specification? Here is the list. So relevant monograph, as we know, for US it is USP, for Europe it is EP, and so on. The next would be the drug substance characteristic. We are talking about DS specification. So I have listed the references for DS. Drug substance, if it is polymorphic, hygroscopic, isomeric, you have to consider those peculiarities, those characteristics in your specification. You should comply with relevant ICH quality and multidisciplinary guideline. Your R&D and analytical research people synthetic research team and analytical research team will list all possible impurities which are generated due to specific process followed. Whatever process is designed, they will list the impurities. And analytical research people will list the control strategy, control plan. That means where to control these listed impurities at initial stage or at intermediate level or at final drug substance level. That control plan would be prepared by research people, and based on this specification would be finalized. Very important point. And along with this, your customer who is expecting you to follow some additional tests, for example particle size distribution, or if his formulation is injectable, then drug substance should have some specific test like microbial enumeration test or bacterial endotoxin test. So that kind of specification, that kind of test should be added in the drug substance specification. So it's not only monograph compliance, friends, but we need to comply with other points also. And then we will have several tests added in the specification, and the specifications are well justified, and that set is presented to regulatory authority. And that is very interesting to have understanding about specification, and which we will be uh, learning when we will join IPM e-learning program. Thank you so much, and I wish you good luck for your uh, plan to take a decision to join IPM PGD RA course. Thank you. Bye.